Good day everyone. So today I'm going to report the DNA replication. First, I'm going to discuss what is DNA replication. So DNA replication is the process by which DNA makes a copy of itself during cell division. The first step in DNA replication is to unzip the double helix structure of the DNA molecule. As you can see in the image, the DNA replication occurs first is to unzip the double helix structure of the DNA. This is carried out by an enzyme called helicase, which breaks the hydrogen bonds holding the complementary bases of DNA together, adenine with thymine, cytosine with guanine. The separation of two single strands of DNA creates a Y shape called a replication fork, as you can see in the image. The two separated strands will act as templates of making the new strands of DNA. One of the strands is oriented in the 3 to 5 direction towards the replication fork, so as shown in the image. This is the leading strand. The other strand is oriented in the 5 to 3 direction away from the replication fork. This is very much evident in the image. This is the lagging strand. As a result of their different orientations, the two strands are replicated differently. Now first, I'm going to discuss the leading strand. So a short piece of RNA called a primer, produced by an enzyme called primase, comes along the binds to the end of the leading strand. So the primer acts as the starting point for DNA synthesis. So the DNA polymerase binds to the leading strand and then walks along it, adding new complementary nucleotide pieces, adenine, cytosine, guanine, and thymine to the strand of the DNA in the 5 to 3 direction. This is sort of replication is called continuous. As you can see in the image, there's a word continuous. So this replication again is called continuous. Yes, it's continuous. Since I'm done talking about the leading strand, so now I'm going to talk about the lagging strand. So what happened in the lagging strand? So numerous RNA primers are made by the primase enzyme and bind at various points along the lagging strand. Chunks of DNA called Okazaki fragments are then added to the lagging strand, also in the 5 to 3 direction. This type of replication is called discontinuous, as the Okazaki fragments will need to be joined up later. Once all of the bases are matched up, adenine with thymine, cytosine with guanine, an enzyme called isonucleic strips away the primers. The gaps where the primers were are then filled by yet more complementary nucleotides. The new strand is proofread to make sure there are no mistakes in the new DNA sequence. Finally, an enzyme called DNA ligase sells up the sequence of DNA into two continuous double strands. The result of DNA replication is two DNA molecules consisting of one new and one old chain of nucleotides. This is why DNA replication is described as semi-conservative. Half of the chain is part of the original DNA molecule. Half is brand new. Following replication, the new DNA automatically winds up into a double helix. That's conclude my report on DNA replication.